Do the animals that feed on sagebrush influence the quality of sagebrush? So she, with Chelsea, she worked with Chelsea on a clipping study to mimic browsing by sage-grouse to see whether or not that actually influences the plants, and that project is also in progress right now. In addition, she did tons of outreach. So she started our Idaho Watch, which is to bring teachers in the local community out to the field to do real research with us. And so she was sort of her and Jordan were the first group that coordinated this event to try and get high school teachers and students engaged in local research and conservation. She was from the Al Dufty Travel Award to go to Iceland, where she put on and shared this new technology called an electronic nose that detects volatiles in plants, and she shared this with an international community, and people were really amazed by how Marcella is. Do the animals that feed on sagebrush influence the quality of sagebrush? So she, with Chelsea, she worked with Chelsea on a clipping study to mimic browsing by sage-grouse to see whether or not that actually influences the plants, and that project is also in progress right now. In addition, she did tons of outreach. So she started our Idaho Watch, which is to bring teachers in the local community out to the field to do real research. With Chelsea. She worked with Chelsea on a clipping study to mimic browsing by sage-grouse to see whether or not that actually influences the plants, and that project is also in progress right now. In addition, she did tons of outreach. So she started our Idaho Watch, which is to bring teachers in the local community out to the field to do real research. With Chelsea. She worked with Chelsea on a clipping study to mimic browsing by sage-grouse to see whether or not that actually influences the plants, and that project is also in progress right now. In addition, she did tons of outreach. So she started our Idaho Watch, which is to bring teachers in the local community out to the field to do real research. With Chelsea. She worked with Chelsea on a clipping study to mimic browsing by sage-grouse to see whether or not that actually influences the plants, and that project is also in progress right now. In addition, she did tons of outreach. So she started our Idaho Watch, which is to bring teachers in the local community out to the field to do real research. With Chelsea. She worked with Chelsea on a clipping study to mimic browsing by sage-grouse to see whether or not that actually influences the plants, and that project is also in progress right now. In addition, she did tons of outreach. So she started our Idaho Watch, which is to bring teachers in the local community out to the field to do real research. With Chelsea. She worked with Chelsea on a clipping study to mimic browsing by sage-grouse to see whether or not that actually influences the plants, and that project is also in progress right now. In addition, she did tons of outreach. So she started our Idaho Watch, which is to bring teachers in the local community out to the field to do real research. With Chelsea. She worked with Chelsea on a clipping study to mimic browsing by sage-grouse to see whether or not that actually influences the plants, and that project is also in progress right now. In addition, she did tons of outreach. So she started our Idaho Watch, which is to bring teachers in the local community out to the field to do real research. With Chelsea. Maximize their dietary quality because it has population level impacts. So due to all of these organism level and physiological an example of some of the maximize their dietary quality because it has population level impacts. So due to all of these organism level and physiological an example of some of the maximize their dietary quality because it has population level impacts. So due to all of these organism level and physiological an example of some of the maximize their dietary quality because it has population level impacts. So due to all of these organism level and physiological an example of some of the maximize their dietary quality because it has population level impacts. So due to all of these organism level and physiological an example of some of the maximize their dietary quality because it has population level impacts. So due to all of these organism level and physiological because it has population level impacts. comparing the grouse to the random plants. And then the opposite selection is true for toxin concentration. They chose the lowest toxins and the highest, or had the lowest toxins compared to those available at random. So we wanted to, again, look at all of this in a different landscape. So we looked at a mixture of three-tip sagebrush and then Wyoming sagebrush. So Wyoming sagebrush is available across most of the greater sage-grouse range, and so that's why it's really important to understand how they make decisions there. And then we also wanted to do some work in three-tip sagebrush because we don't really know very much about it to start. And then it's also one of the few species of sagebrush that has a range that's expected to expand in the future. So it has the ability to resprout after fire, which a lot of sagebrush species have to reestablish from seed, and so it kind of has a competitive advantage and the ability to increase after fire, which is important in the Great Basin. And then it also is likely to increase its range based on temperature. So because of all of this, we saw that it is likely to be important in the future, but we don't really know anything about how animals respond to it. So there's very, very little research on wildlife or domestic animals using three-tip. So previous grouse literature has said that grouse will select for Wyoming sagebrush over three-tip for nesting cover, but and then if they do select three-tip, they have lower nest success, but that's really about all we know. So it's important for us to understand how grouse might use it in the future. So going back to this toxin dose response, our response here is going to be the behavior of the animal. And so our question was, do toxins influence selective foraging as a behavior? And our hypothesis was that they should. So animals should try to minimize their toxin intake and maximize their protein intake at multiple spatial scales. And so for those multiple spatial scales, we're looking at between plants and then also between patches and at a landscape scale. Our study site was at Craters of the Moon in Idaho, so we're kind of on the southern end. And in this map here, you can see all of these black areas, which are lava fields. And then all of the sagebrush has these polygons on top of it, and the polygons are provided by BLM, and they show fire. So the more red the polygon is, the more recent the fire. So these dark red ones were in the last 10 years, and then all the way back to these kind of light yellow ones, and those were in the last 30 years. So based on where the points fall, you can see that every plot had been burned sometime in the last 30 years. And so this has resulted in relatively low sagebrush cover. So this is kind of what the site looks like. It's about 7% cover, which I'll get into more in the discussion, but that's pretty low. And it includes both three-tip and Wyoming sagebrush, but it's not a perfect mix. So at some patches, one species is available, and at some they're mixed. So I'll show more about that later. But to perform this research, we follow birds with radio transmitters on them. So we approach flocks using radio telemetry, and then can identify their patch that they were foraging in using pellets and tracks when snow is present. Otherwise, we just use pellets. And then you can identify plants that they've browsed on, because grouse will clip off leaves, and so it leaves kind of this sharp leaf margin. So it's actually pretty evident which plants they were actually selecting. And you can then measure the intensity of browse based on the count of the bites. But then you can also just do a class of browse or non-browse, which is what my analysis used. And then we also measured structural characteristics of the site, so that's percent cover and density and height of all of the plants. And then we repeated all of that sampling at randomly selected points. So these were points that we put into ArcGIS, constrained by the boundary of known winter grouse habitat in the area. And then we took the plant samples inside and then ran all of these different analyses on them. So the one that we don't do in-house is protein content, so we send that out to New York. And then the rest we do in-house, so I'll show a little bit more about them. This is headspace gas chromatography, which uses a headspace autosampler and a gas chromatograph. And it's designed, the method we use is designed to identify monoterpene compounds. And so this is a typical chromatograph printout. This is for a cocktail, so it is used to identify known compounds. And the height of each peak is the concentration, and then each peak is an individual compound. And then each compound comes out at a different retention time, so that's how you can identify unknowns in your samples. So then the last two assays that we do are phenolics and coumarins, and this on the right here is an example of the coumarin assay. These are plate reader assays or colorimetric assays that measure color intensity, which is correlated to the concentration.
So we have 2 different assays for coumarins, which is fluorescent, and then also for phenolics, which is a fast blue assay. And then briefly for statistics, we did, to compare between the 2 species, we did a univariate non-parametric test, and then landscape scale analysis was a chi-squared. And then for patch and plant scale selection, we used logistic and conditional logistic regressions, respectively, and then used AICC model ranking to compare among all of the dietary and structural predictors. So for the species comparison, to start, I just kind of want to go over, this is a chromatograph that has an overlay of Wyoming big sagebrush and of 3-tip sagebrush. So Wyoming's in top in red, and 3-tip is on bottom. And so the first thing that you might notice is that 3-tip sagebrush has really, really tall peaks. So its concentrations of monoterpenes are about 1.5 times higher. So it's a huge difference in toxin concentration. And then the other thing that might pop out, it might take a little bit of counting, but 3-tip sagebrush also has fewer compounds. So Wyoming has about 1.5 times as many. So it has lower concentrations, not just overall, but also per compound. Oh, and then also there are little asterisks, and those denote compounds that are specific to that species of sagebrush. And then not pictured, 3-tip also has higher coumarins and lower total phenolics. So based on all of this, because Wyoming has much lower toxin concentrations, it seems like that would be a better food source for grouse. Then we also looked at protein content, and again, Wyoming has a higher crude protein, which makes it, from our perspective, a better food resource for grouse or any wildlife. And then our final comparison was in structure. So dietary quality is really important, but structure can also play a big role in where animals spend their time. So we looked at plant height and found that Wyoming was quite a bit taller. And this greenish bar that I have shows the recommended habitat guidelines. And those are around 25 to 35 centimeters. So 3-tip falls within that range. But you can see that Wyoming is quite a bit taller. So this is one of the big differences between the 2 species. So we took all of that information and then tried to see if that influences landscape scale selection. So I'll kind of walk down from landscape scale to small scale selection. But at the landscape scale, we didn't really see any selection occurring. So this is the same kind of table as before. So we have our habitat types of Wyoming sagebrush, 3-tip sagebrush, and a mixture of both. And you can see that the columns for used and random match pretty closely. And so it kind of suggests that they're not really selecting one sagebrush species over another. So that was kind of strange to us based on having those huge, huge differences in the chemistry. But we've still looked at small scale selection. So this is at a patch scale, which is looking at why grouse select one patch over another. So this is comparing between those used patches and the random ones. And we broke out our analysis by species because of those huge chemical differences. So with Wyoming sagebrush, this is looking at all of these models were selected using AICC, and then we went through and removed models that had low reliability. And so these are the top 2 models for Wyoming, and the 2 big differences are that random plants were quite a bit taller, so this suggests grouse was selecting for short plants or shorter, and then the average concentration of unknown 21.0 was also lower in used plants. And then for 3-tip, we only had one model that performed particularly well. And it was again plant height, and so we saw that grouse were selecting for those shorter plants for 3-tip as well. Then at plant scale selection, this is again between a browsed plant and a non-browsed plant within a single patch. So this is kind of ignoring all of that available at a landscape level, so all of those random plants. And again, we broke it out by species, so this is Wyoming sagebrush, and we found that browsed plants were both shorter and then had lower concentrations of toxins. And one thing I want to point out with the plant height is that the browsed plants were shorter, but they still are mostly above the recommended winter habitat guidelines. So they're still quite tall, but they're shorter than other ones that are available. And then having lower concentrations of toxins is something that we would expect to see as well. And then at the plant scale for 3-tip sagebrush, we found that browse plants had higher protein content, which is again something we would expect to see, and then higher numbers of monoterpenes. So this is if you go through that chromatogram and count out the number of compounds. And so this seems a little bit contrary to what we would normally expect because it's a higher amount of monoterpenes. And so this suggests that there might be some benefit to consuming a high number of these compounds rather than high concentrations. So then we took all of those plant scale models and we went through and evaluated how reliable they were, and then used the two most reliable ones to do this generalized additive modeling. And so the way to look at this is the bottom is the average value for the entire catch. So it's averaging both browsed and non-browsed plants. And then on the y-axis is the difference in plant height between the browsed and the non-browsed plants. So anything at 0 shows that there's no difference between that parameter for browsed and non-browsed plants. Anything above shows that they were selecting for that parameter, and then anything below is selection against. So basically, the take-home for this entire graph is that there's really no selection in plant height until you get to about 55 or 65 centimeters, and at that point, grouse are selecting for shorter plants, which mirrors what we saw with Wyoming sagebrush quite well. And then we also did this with unknown 21.5, which is a monoterpene. And it kind of shows a similar pattern. There was kind of a threshold a little bit later, and that was about 10 AUC per 100 micrograms, which is basically just a unit for measuring concentration of toxins. And at that point, grouse started to select for lower concentrations of the toxin, which is, again, kind of what you would expect based on previous foraging literature. So all of this led us to the question of why are grouse not selecting for Wyoming sagebrush at a landscape scale? Because at small scales, they're selecting for the same traits that we would expect. So they're looking for lower concentrations of chemicals and higher protein, and so all of these things seem like they would make it make Wyoming a good foraging resource. But since we didn't see that, we looked at 3 reasons that they might not have been. So one is that it could be a landscape scale relationship that's driven by chemicals that we didn't measure. So like I mentioned, sagebrush has hundreds of chemicals and we only measure a subset. So it might be something related to that. It could also be related to height. So plant height is important for animals because it protects both from predators, but then also their ability to see approaching predators is reduced by having tall shrubs around them. And then it can also influence the microclimate, which is important. There are some papers with ruffed grouse that show that microclimate selection is really, really important for those species because they are just out in some pretty harsh conditions throughout winter. So it could be sagebrush height, and then the other possibility is that the sagebrush cover at the site is really low. So I mentioned that at the beginning. So you have a picture on the left of craters and then on the right of Raft River. So craters is about 7% covered. The Raft River is about 23% in this area. So that's a pretty big difference in selection, or I mean in cover. And previous sage-grouse diet selection studies have had cover between 17 and 40%, so that's quite a bit higher than the 7% that we had. And then also the winter habitat guidelines are between 10 and 30% cover, so again, craters is pretty low. And so from a foraging perspective, having a lot of cover might represent having a diversity of choices, so it might be the difference between shopping at a gas station convenience store versus Whole Foods or Winco or something like that. You have a lot more options to make choices at those big grocery stores versus a gas station convenience store. So you might make different foraging choices at those 2 spots. So our management implications, winter habitat for grouse is pretty limited. So this map shows the current range and historic range of sage-grouse. So sage-grouse only occupy about 56% of their pre-settlement range. And then from that, in some areas, grouse use only a fraction of their habitat available for winter. So it's really important to have both high quality and quantity of sagebrush available. And unfortunately, in the Great Basin, there's a high fire risk. And so this might make it likely that there's going to be continued reductions in both quantity and quality of habitat. But now we know that grouse do eat 3-tip sagebrush, but we don't know if it's providing an optimal food source or if it's something that grouse eat and find acceptable. So future studies could possibly evaluate if it's something that grouse would eat in a pre-fire or pre-disturbance habitat. But to sum up, we did see that there was, at some scales, there were influences of toxins on foraging behavior. So then our next question was how toxins influence physiology of animals. So looking at parasite loads. So we thought that toxins should influence parasite loads based on 2 different hypotheses. So I'll walk you through those. The energy constraints hypothesis is that animals experience a trade-off between parasites or immune function and detoxification of toxins. So in this, this would kind of relate to that caterpillar example that I showed earlier. So animals with high toxins in their diet may not be able to invest as much energy into immune function, and therefore might have high parasite loads. And then the other alternative is the self-medication hypothesis. And in this, these animals are consuming high doses of toxins, which their intestinal parasites experience and are exposed to, and then those toxins can regulate the parasite loads, so animals would have low parasite loads correlated with high toxin loads. So we have a couple examples of animals using toxins for medicine, and all of the examples currently in the literature are of generalist herbivores and domestic animals, so I'll go through an example of each of those. And the first study was with chimpanzees, and it was purely observational. Researchers noticed that some chimpanzees were consuming plants really high in toxins when they were infected with intestinal parasites, and then as their infection decreased, then those animals would switch their diet back to a normal diet that had low toxins. So the key to success for these animals is that they have to have a high toxin excretion and low absorption. So that means that the toxins are going through their intestines unchanged, and that the parasites then are experiencing high toxin loads, but the hosts, because they're absorbing and metabolizing relatively few toxins, should have lower physiological impacts on the host. The other example was with domestic sheep. So previous research found that domestic sheep will self-medicate with tannins, which are a class of compounds, and that they would discontinue their use of tannins when parasite loads had decreased. So they would treat sheep with their banks and then see if they would continue their tannin-rich diet. So these are two great examples of animals using toxins, but we don't know how specialist herbivores do with self-medication, if it's even possible for them. That didn't show up well, but supposed to say that individuals have population level impacts of parasite loads. So here are three examples of animals that have impacts on both physiology and mating success. So with sage-grouse, they tested males with malaria and with lice, and animals that had high parasite load or pathogen loads had low mating success, and then they took some of those animals into a captive environment and treated them, and then those animals had better mating success than males that didn't get treatment. And then other examples are with guppies and with Grant's gazelle, and in both of those species, males with high parasite loads have a trade-off and have lower reproductive success. And then it also scales up to have population level impacts. So this has been shown with both willow grouse and pictured here is the red grouse. And in both of those species, parasite populations can drive populations of the entire species of grouse. So this is a graph that shows the number of grouse in the population related to time. And all of these different cycles are parasite driven. So these here are in a natural population with no treatment. And then as the proportion of treated individuals increases, you get to this line, which is 100% of the population being treated. And at that point, the cycle stops. So this kind of helps support evidence or give supportive evidence that parasites can influence. So it's really important for animals to be able to regulate parasite loads, both from an individual and a population level standpoint. So then also we were looking at if specialist herbivores have the ability to self-medicate. So specialists are already at the upper threshold of their toxin intake, and they have low tolerance for novel toxins. So while generalist herbivores can consume a wide variety of toxins, specialists have a pretty narrow dietary niche, and so they may not be able to consume the new toxins that could help regulate parasites. And the reason new toxins are important is that a lot of these specialists may have parasites that are resistant to the toxins that they normally consume because they've been exposed to it over evolutionary time and may not be able to regulate parasites using the toxins that are normally part of their diet. So we again looked at this with greater sage-grouse. And the reason, first of all, they have a highly specialized diet, so that's important. But they also have this abundant tapeworm called Regnellus intracerci. It's really common both range-wide and then also in individual birds. And it can limit nutrient acquisition, so that can influence the energy available for both immune function as well as for toxin detoxification. So the other reason that this is a really great system to test this in is that we know sagebrush has some anti-helminthic properties. So previous research has tested this in vitro on parasites from both generalists, or from only generalist herbivores. And they found that individual monoterpenes, including cineol and camphor, which are common in sagebrush, as well as sagebrush extract, can have antiparasitic properties. So that would make it possible then that animals could use this for self-medication. And then the other important factor is that there's low toxin absorption in grouse. So this is another graph that shows this grouse species in red on top, compared to sagebrush leaves in blue on the bottom. And all of the peaks match up, so they're excreting these monoterpenes unchanged. They're not metabolizing them, which means that those are going through their gut the entire way, and that that's dosing parasites. And the other important thing here that I didn't picture, but some people in Jen's lab had tested to be sure that those toxins are the same throughout the intestine, so pulling out pieces from each section of the intestine, and what comes out in the feces is the same as what's in the intestine. So we know that if we use fecal monoterpenes, they're a good proxy for what the grouse are actually, or what the parasites are actually experiencing. So we tested this at 4 study sites throughout Idaho. I'll talk about both the regional scale modeling, which is including all 4 sites, and then we also did modeling only at Raft River because it's the richest data set. So for the fecal pellets, we took them into the lab and performed 2 assays. The first was looking at fecal monoterpene content, because that's again exactly what the parasites are experiencing. And then we also did a McMaster egg count, which is a really common technique in veterinary medicine for quantifying parasite loads. And then previous research with red grouse has shown that the McMaster is correlates egg abundance with adult parasite abundance. And so it's a good proxy for how many parasites are actually in the intestines. So our statistics, again, really briefly, but we used log of eggs per gram dry weight as the proxy for parasite load, and then performed a 2-stage modeling approach using information theoretic to help narrow down our ecological and dietary models, and then compared them again using AICC. So I'll first talk about all of the regional scale modeling. So this was again with all 4 sites, and we took only the samples that were collected during midwinter, so kind of around January. And we included, for all of these models, we included site as a covariate because you can see here there's a fair amount of geographic variation. So the top model was fecal camphor with bird sex and site. So this is a graph of fecal camphor and then parasite load on the y-axis. And at high fecal camphor concentrations, we saw higher loads of parasites, which suggests with this chemical that animals are experiencing that energy constraints. And then the next model was alpha-phalendrine with bird sex and site, and it told kind of a different story. So it showed a decrease in parasite loads at high concentrations of toxins. And so although they contrast, I want to point out that fecal alpha-phalendrine, there are a lot of zeros over there on the left-hand side, so it's not present in all sagebrush species, and therefore it's not present at all sites because each site has a different set of plants available. So this is really kind of driven by a single site, by the Owyhee site. And so it's not really a true regional scale model, but it did come up as a top model. And then this was the last predictor that had been paired with both of our top models. And it was bird sex, so males are on the right here and then females are on the left. And males had much higher parasite loads than female birds did. And this makes sense from a lot of the literature. There's the immunocompetence theory, which suggests that males invest more energy towards producing hormones and invest more in reproductive health. And so they had this trade-off, and they can't defend themselves well against pathogens and parasites, so they have higher parasite loads. And then we repeated all of the modeling at Raft River. So this is, again, just, it's a single site, but it includes all of the different parasite loads. Sorry, it includes all the different seasons. Sorry. Anyways, so we continued on with modeling, and we found 3 top models, which I'll show in a minute, and all 3 of the top models for this had high parasite loads correlated with high toxin loads. And so, oh, yeah, for some reason, I'll just shut down all of my PowerPoint stuff. I'm getting it. It's slow. Let's scroll through. But anyway, so all of the top 3 models for the parasite modeling had top models with high parasite loads correlated with high toxin loads. So almost there. Okay. Oh, okay, really? Okay. So now you can actually see it. So our top predictor was fecal sineal, and again, it was had a positive correlation, which kind of supports that energetic trade-off. And then our next was total fecal monoterpenes, which again have that same pattern. And then finally, fecal compound diversity, which again followed that same pattern. So all 3 of those at the Raft River site, as well as the camphor concentration at the regional scale modeling, supported the energy constraints hypothesis. But the other important thing to recognize is that it also shows that these parasites are likely to be resistant to sage-grouse toxins, or I mean to sagebrush toxins, because they're able to withstand pretty high concentrations of these. So we wanted to evaluate this a little bit closer. So Jen mentioned this in the introduction, but we started some in vitro assays. So we took from the Thesis, we cultured goose parasites, also in the Ravenella family, and would test them with all of these toxins in sagebrush. And so these are some of the results from yeast. We haven't done all the grouse stuff yet, but that's the next step, is to see if they respond differently. But with yeast, we found that, so this graph shows the 5 treatment groups, and then the percentage of eggs that hatch. So if there are a high percentage of eggs hatching, then it shows that it's a condition that the egg can persist in. And then at low percentages, it shows that drug is inhibiting their ability to grow and hatch. So levamisole and albendazole are 2 commercially available antihelminthics. And then cineol and camphor on the far right-hand side are 2 sagebrush toxins. And so you can see that cineol and camphor at concentrations that are actually experienced by parasites in sage-grouse deaths match pretty well with albendazole, although levamisole is a little bit stronger, but it's showing that they have some antiparasitic properties against goose parasites. So the next step will be to see how grouse parasites respond. And then probably the most important management implications are that, like I mentioned, a lot of grouse species do have population cycles that are driven by parasite loads, and so it's important to evaluate this over long-term scales. So we have, at any single site, we have a 2-year data set, which is not adequate to evaluate parasite loads, but we didn't see any variation in the 2 years of data. But it's something, again, that should be studied over a much longer time frame. And then the other interesting thing was that I had sage-grouse winter flock size as one of the parameters for testing parasite loads. And we didn't see any relationship between flock size and parasite load, which was kind of, I expected to see that high flock densities would contribute to high parasite loads because that's a common denominator, I guess, in transmission risk. So we didn't see that, and so I think one of the things to look at in the future would be how densities at different times of year impact winter flock sizes. So the transmission of this parasite likely occurs during summer when beetles and ants are available for chicks to eat. And so it might be that flock densities at that time of year impact winter flocks, or impact parasite loads more than winter foxes. So to sum up, we did test toxin dose from both a behavioral and physiological standpoint. And we found that toxins can influence selective foraging at some scales, but not all scales, and that toxins did influence parasite loads in more of an energy constraints relationship than a self-medication relationship. And then here are all of the acknowledgements, and I have a ton of people who are here, which is awesome. So my committee is Jen, Jack, and Jim. And then Idaho Fish and Game, Pittman Robertson Funds, Boise State, UC Davis, some researchers from there. And then we had a ton of really great students that came in and helped from the labs. They have a variety of different funding. And then Sigma's Eye, NSF. And then Fish and Game personnel who are again here, which is awesome. And then 4B lab members. This is a really long list, but they've all contributed so much. It would not have been possible without all of these people. And then we also had, I didn't put them on here, but we also had a number of volunteers that came out for weekends in the field in the cold. So a huge thank you to all of these people. And then any questions?
For the gas diagram, we were looking at the different peaks, and you talked about unknown 21.5 as monotropic. How do you guys know like what that effect is, or even that it's monotropic, just based on other research that you've been with, or where does that fall into place with the monotropics?
Like you mean as far as identification goes, or
Yeah.
So there are a couple of other standards that Brecken had found or had made years ago, and they have some monotropies around that time. But because that cocktail led to a lot of overlap between some of the identifying compounds, it's not included here anymore. So to me, I have no idea what I think it is, but we need to like test it individually to really be sure that it's in that range. But I think with Graham's work, he had cut off around 24 and found that most monotropies come out before then, so it should be in that range. Any other? Okay.
So they're obtaining the parasites via insects, mainly, or could they be getting some from vegetation itself as before?
Yeah. So nobody actually knows with this species. Great melon is really common in game birds, so most of the research has been with pheasants. And with pheasants, it's transmitted, so it's released in the feces, and then ants and beetles pick it up, and then the chicks eat the ants and beetles, and then that's how they get the parasite loads. And so I think that's part of why I didn't mention those technological issues, but threw me off. But in the fall parasite loads, there's a ton of variation, and so I think that's probably because that's when those young grouse are picking up a lot of that, a lot of the insects at that point, and then the adults are probably staying pretty level with where they've been all year. So that would be what we think it's happening.
That parasite persists then in the individual grouse through the year. Or is there a point in time when they are free of it and then they get inoculated again?
So the parasite loads do kind of show with ours anyways, and what's kind of suggested with Fremgen, they do kind of cycle. So we don't know what happens this summer, but they do have a little bit of cycling going on. And so again, kind of what I think is happening is in fall you have the juvenile birds that have really high parasite loads, and it has been roughly shown in like the 1950s they thought that was the case, and then adult birds had lower parasite loads. And then in midwinter, I think they all just kind of switched to the same diet, and they have similar physiology at that point, so it makes sense to have relatively low variation. And then I think the jump in spring is related to investing into reproduction rather than to host immune function. But then on the flip side, I gave like all of the host reasons, and it could also just be that that's the way the parasites like this. So, Roger?
Do you think the shorter plants are more preferred, or the shorter plants are shorter from being browsed by grouse?
Yeah. So at Creighton's, I did some tests to see if there were differences in like chemistry overall. I didn't really see any major, so I think it was mostly just that, and I guess also in the field, I don't really remember seeing any signs of like mule deer grouse or pronghorn grouse or anything like that. So I think it could just be a structural characteristic that they like, but then also the thing that I didn't really talk about much that Jen and I have kind of been looking at with the Raft River site is that compound diversity might be really important for animals to maximize for a variety of reasons, but it might be beneficial for animals to consume both Wyoming sagebrush and fruit sagebrush, and so that might kind of funnel them towards selecting more of that free tip, or selecting some of that free tip to kind of maximize that diversity. And so then the height is just kind of correlated with that species selection. Yeah. Well, but yeah, also just thinking, because you know a lot about that Raft River site, and at Raft River we are kind of seeing that the small are really preferred more than the medium at that site. So yeah, so sort of background. Any others?
So I remember there's, and I can't remember which way it is, but some males travel farther away from the leks and sprinklers.
Yeah.
Do you think closer to the lek there's more likelihood of picking up parasites? Farther away there could be less?
Yeah, that's a good question. I think with the, at least with the present research with that genus of parasites, they correlated it to density of ant mounds. So I don't know. I guess it would depend on the spatial configuration of that. But we didn't see one of the things that came out. It wasn't like as good a model as some of the others, but elevation came out as a decent model. I thought that was kind of interesting because it was higher parasite loads at higher elevations. So there may be some geographic patterns that didn't come out in our top models, but may be important also.
Okay, any other questions? Thank you, Marcella. Before you go, anybody who wants to come and celebrate or congratulate with Marcella after tonight, I'll write my address up on the board. Many of you got the email. You're welcome to come, have a drink, celebrate with us. We'll be at my house at 6. I have to kick you out by 10 because my neighbor doesn't like my parties at my house. I think I'll have to get up and do things the next day. So please come and celebrate with her. So I'll write the address.
Well, I just, I mean,
Good.